and good morning, learners, teachers, whoever has joined the session in addition. And we basically want to give a little background. I'm not going to be long because I don't want to take a lot of time around the activities, the activity itself rather. These are the activities that we normally would do, you know, here at Itemba Labs in face sort of contact in trying to motivate learners that are studying mathematics and sciences, where we would bring on board professional, practicing professionals in the field so that they can come in and motivate the young ones and trying to show them that there's light at the end of the tunnel. In fact, the future is normally bright when you do these subjects. And we tend to try and choose our speakers carefully. We try and look for people that will sort of, you know, be able to resonate or connect, you know, with the young people or young learners, and also people that are doing work that also sort of is in line with the challenges and the issues that people in the streets or everyday people are facing, like, for example, the present crisis of the coronavirus and other issues that normally are a challenge to the public. Hence, we, for example, specifically this time have invited Dr. Hlumani Ndlovu from the Faculty of Sciences at UCT. And so the intention is that the lecture, because we have to work in this format now, where you cannot come physically to one space and interact with the speaker, the format is such that we have to do it virtually, and unfortunately the technological glitches are always there when we do things this way. So without taking any of your time, I will hand over to Dr. Hlumani, perhaps to say a word or two. Also, before I forget, we will work this way. We have a number of schools, that about 4 schools rather to be exact, that are joining us, and we have questions here that you have all sent that you would like Dr. Ndlovu to respond to. And so what we will do, we will choose your school and you will then get a chance to ask the question. And the format is that we will have round 1. In other words, round 1, you will have your 3 or 2 questions that you were given that you will be asking, and then we'll move to the next school and then to the next school until we have all 4, and then we come back for round 2. And if there's extra time, we go to the other questions that you couldn't ask. And so without further ado, I will start with what my page tells me to start with here, which is Masibambisane. Is Masibambisane in the house? In the room? Okay. Okay, it's fine. If ma'am, okay. Ma'am, you are first on the list here. Are you ready to ask your questions? Does anybody, or is anybody asking questions from your side there, or should we read questions ourselves? Because we've got the questions here.
Yes, they are ready. Yes, they are ready.
Okay, get somebody to ask your first question then. Okay.
Good morning, doctor. I would like to ask, when a scientist discover a vaccine, how do they make sure that it is not harmful to people?
Yeah, I heard the question. Can you guys hear me? Sure. Thanks. I mean, thanks for the question. Are they going to ask all the questions at once and then I answer all of them?
Yeah, maybe let's do it that way.
Yeah, okay.
Give us your second question and the third question so that we can get all of them at once and then we move to the second.
Okay.
Yeah,
There's a second
okay.
question.
It will work better that way.
Okay. Okay. Second question.
Good morning, doctor. I want to know if you're sick, can the vaccine help you?
Okay.
Okay, last one. Third one.
Greetings to you, doc. I would like to ask you if you are allergic to a vaccine, what happens to your body?
Okay.
I'm gonna respond the school because there's a lot of noise in the background. Yeah, sure, perfect. Thank you. Good morning, everyone. As I have been introduced, my name is Hlumani Ndlovu, and I work at UCT as a lecturer. Thanks for the opportunity again once more this morning to interact with the learners. Thanks to Lucretia and Hilton for organizing this session. It's an honor for me to get the opportunity to interact with the young minds that we have in the country. And I see that we have a lot of very, very good questions, very difficult questions. So I'm going to try my best to answer all the questions. So the first question from Masibambisane was, how do we check the safety, basically, of the vaccines? So just to maybe give a little bit of background in terms of how vaccines are developed, because this is a very thorough process. And I guess the question with COVID vaccines is why people are concerned about their safety is because of the speed with which they got to market. We have a lot of vaccines that we take, particularly in this country. As a child, you take a lot of vaccines. So vaccines is part of what we do. In fact, in terms of dealing with disease, you think about elimination of polio. We were only able to eliminate polio in the world because of vaccination. So vaccines are very good for us. And they offer a lot of public health benefits in terms of preventing the spread of disease. So when we develop vaccines in the lab, one is, of course, you do the work of identifying which component of the virus or the bacteria that you think is immunogenic, right? That you think will cause an immune response in a human. And in the case of COVID-19, what they've identified as an immunogenic component is what is called a spike protein. So if you think of a virus as this round, a pathogen, and then on the surface of this virus are these proteins that basically, extend outward, and they're called spike proteins. And these are the proteins that allow the bacteria to bind into target cells, and then it's able to infect human cells through binding via what is called your ACE receptor. So this is a receptor that will basically bind this spike protein and then allow the virus to enter our cells and then be able to propagate itself. So, and this is a component that is used in most vaccines. Like, for instance, the vaccines that we're using in the country right now is the J&J vaccine. And part of the component of that vaccine, or part of the, what they use as a virus component is this spike protein that we then trying to make an antibodies against the spike protein to prevent it or to prevent the virus from being able to gain access to our immune cells. And this is how then we're stopping the infection. So now, once you've done that part, which is to identify the component of the virus that you want to include in your vaccine, then now you're going to test for efficacy. Does this component actually does what we think it does, right? Does it induce an immune response? So typically what we'll start with animal models, specifically your lower order animals like mice. So you take mice, you then inject them with this vaccine, and then you look at the immune response. do you get T cells, B cells, responses, and all that stuff. And then if you see that you're getting those responses in mice, you then move into higher animals. In most cases, they use non-human primates, which are monkeys. And this is because evolutionarily, they are very closer to humans, right? And again, here, you want to check the safety, you want to check the dosage of how much should you give of this vaccine to induce a robust, strong immune response. And then if you see that, again, when you go to high other animals like monkeys, you're getting a very good response, then you then go to humans, right? So before you get to humans, you still have a lot of steps that you need to go through to prove that this vaccine, this component of a vaccine that you are using to induce immune response, does it do what it's supposed to do, right? And then you go through the monkeys, you pass the stage, everything is going very well. Now, when you get to humans, it's very, very strict. Clinical trials are highly, highly regulated because, again, you don't want to cause harm to humans, right? There's a principle of doing anything with human beings that you don't want to cause unnecessary harm. So safety is one of the key things that you want to check. So the first thing is that clinical trials go through various phases. You have phase 1, phase 2, phase 3, right? Phase 1 is this is where you're checking for safety. So you're taking a small sample of people, a small number of participants. You then give them, different doses of your vaccine of interest, and then you see whether they develop any reactions to the vaccine, right? So this is here you're looking for safety. Do they develop flu? Do they have allergic reactions? What happens to them, right? And then if you find that in that sample or in the cohort of patients of people that you have recruited, they're not showing any problems, then you go into your phase 2 clinical trial, which is where you're looking again. Now, this is a bigger sample size, so we have now more participants in your study. And then you're looking at two things. You're looking at, again, safety, but also now you're looking at immunogenicity. Does the vaccine induce the immune response that you desire, right? And again, if you can prove at the stage that this vaccine, 1, is safe, 2, is immunogenic, then you go through the last stage, typically, which is a phase 3 clinical trial. Now, in phase 3, you're looking at one thing specifically. You're looking at efficacy. And this is where also you include the control, what is called the placebo control, where you have people in that random pool of participants who get what is called basically a dummy vaccine, which could be just like, getting an injection with water, for instance, without having the active component of the vaccine. And then you get the people who then get the real vaccine, and then they go on and live their normal lives. And then you see if those who got the vaccine, do they get infected compared to those who don't get the vaccine? Then this is where then you can say, okay, fine. Out of 1,000 people who got this vaccine, only 5 got infected. Compare that with the 1,000 people who got the placebo, and only 100 got infected. Then this is when then you say, okay, fine. This vaccine is 60% or 90% or whatever percent effective, because when you compare the two arms, those who got the vaccine against those who did not get the vaccine, how many of them were infected? And this is how it works. So at every stage of the development process, one of the key things that we check for is safety. Is the vaccine safe for humans? Because if a vaccine causes serious complications, there's no point. It can't go into it can't be approved, right? Part of the approval process is to show that the vaccine is safe. And this is why, again, with the COVID vaccines, although the processes were rushed to an extent, but they had to do those clinical trials and get the data to prove that these vaccines are safe. And this is really how it works. And this is how we try to avoid giving harmful vaccines to humans. And again, this is true not only for vaccines. Again, if you want to develop drugs, let's say you want to develop a new drug for cancer, it's going to go through the same process. Going to go through the preclinical models like mice and monkeys before it gets into humans. And once it gets to humans, it's going to go through the different stages of clinical testing before it gets approved. So that's a response to the first question. Second question is, if you're sick, can a vaccine help you? Well, ideally, what vaccines are supposed to do is they're supposed to prevent infection, right? This is what we're using the vaccine for. We want to educate the immune system to see parts of the real deal, part of the virus or the bacteria before you getting the real infection. So by the time that you're getting exposure to coronavirus, your immune system has had exposure to the virus. and then is more equipped to quickly respond and deal robustly with the virus, and then deal and clear the infection. So that's what vaccines are there for, right? They're not there to basically prevent, they're not there to offer therapy, but they're there to prevent infection, right? So what we call POR, prevention of infection. It can, I mean, in the context of COVID-19, in terms of the vaccines that we have, because none of the vaccines that we have been shown to prevent infection. What they do is that they prevent people from developing severe disease. So meaning that, and this is partly why also with COVID-19, although you may get vaccinated, you're still required to wear a mask because you can still get infected even though you had a vaccine. It's because the vaccines don't prevent infection, but they prevent you from developing severe disease, right? I see that there seems to be communication issue with Ms. Mamputa. So I'm not sure if I should wait or should I continue with giving a response. You may help me here. Should I wait for them to have a connection or should I continue?
I think because of time, Doc, I think you can continue, and then perhaps when the break time is off, then they can hear you well, because it's something perhaps that we cannot control at the moment. They're having a break time. Yeah.
Okay, perfect. Yeah, so just to continue from there, so really that's what the vaccine is for, is to prevent infection. Now, if you're already sick, what you should do is to get therapy, right? And this is where now your antiviral medication comes in. Like, for instance, I mean, there's many drugs that's been tried and tested for COVID-19, and a lot of them have been proven to be ineffective. Like, for instance, there was a huge noise in this country, in SA, about a drug called ivermectin, where doctors were calling for the approval of ivermectin for COVID patients. But there's been many trials done overseas which show that ivermectin is not effective against COVID-19 or coronavirus. And there's many others. I mean, I can think of, there's been studies looking at, there was this drug, I mean, there's a big noise in the US called chloroquine, hydroxychloroquine, where they were saying it's been used a lot for malaria. So if you live in Africa, you'll be aware of malaria, which is a parasitic infection, which is very lethal. And we've been using, chloroquine to treat people with malaria. And they were using it in the US for COVID. Again, that one has been shown to be ineffective with coronavirus. And many others. I mean, like, the list goes on. I mean, there's been so many trials, and I think this is now beyond reasonable doubt, where scientists have been able to show that these drugs don't seem to offer much of therapeutic benefit. What I've noticed in people with COVID-19, that some of them actually, what they develop with severe COVID-19 is that they develop what is called your almost like getting liquid in your lungs. It's a very inflammatory response. So what they typically do is that they give some of these COVID-19 patients what is called steroids. Steroids, basically, they're broad-acting agents that try to basically minimize inflammation. But also what they've seen as well is that some of these patients tend to develop what is called blood clots. And this is, the mechanism I'm not going to go into. And what they do also is that they tend to give drugs that try to prevent the formation of blood clots, just so that a person can have a chance for their immune system to fight the virus and not die because your veins get clogged and then you die of blood clotting, and all those problems. So if you're sick, really you need to get therapies. Vaccines are there to prevent infection. They're not supposed to offer therapeutic benefit. The last question about allergic responses to vaccines. Again, we don't know how our body will react to anything that is foreign, right? So somehow the body can react by making this, a very strong immune response against this foreign antigen, this foreign, vaccine that you are trying to use it to help the body to deal with future infections. And if a person is shown to be allergic, typically, let's say, for instance, if you think about allergies, like food allergies, if you're allergic to what you eat, you stop eating the thing that triggers an allergic immune response, right? In the case of a vaccine, we don't need to manage the symptoms, right, of that allergic reaction. because you already have had the injection. So the best thing to do is to manage that reaction. I actually had a friend who is in the US right now, and she got a COVID vaccine in the US, and she was in hospital for 2 weeks because she developed a very severe allergic reaction to these COVID vaccines. But I think it has to do with probably her immune response, because what had happened is that she took a flu vaccine before that, and she had moved from SA to the US. So there was a change in her body had to adapt to the new environment. And that just made her a little bit, susceptible to these kind of allergic reactions. But the best thing that you can do when you develop an allergic reaction is to stop taking what is causing that reaction, and then allow the body maybe taking like steroids to almost lower the unintended inflammatory response. Because basically this is when your body is trying to almost, the way that comes to mind is your body is overreacting to something that it should not overreact to, right? This is basically what is an allergic reaction, that you have this uncontrolled immune response that then, tends to then cause this, either like you have itchy skin, or you have these patches on your skin of like, almost like, when you're getting bitten by something and then you've been itching and scratching, that then it becomes like discolored your skin. And this is an indication of an allergic reaction. So the best thing you can do there is just to stop, what is triggering. If in a context of vaccines, is to give something that will lower the inflammation and then wait it out.
Okay. So. Thank you. We have Nomzamo next in line to ask their questions. Are you able to ask questions, Nomzamo? Hi.
I also have questions on my side. So I mean, if are they available, Nomzamo?
Nomzamo, are you available? Okay. If Nomzamo is not available for now, we'll move on to Hector Petersen. Okay. All right. So Hector Petersen, if you are available, it's your turn to ask your questions. Hector Petersen.
But I mean,
like already.
Okay.
Because we're struggling with the communication.
So you can't hear us.
Struggle to connect because the Wi-Fi is utilized by the learner. That's a problem. Now it kicks out all the time we get in, but we are going to try by all means.
All right.
Because our IT guy is here trying to solve the problem.
Okay. All right.
All right.
So in the meantime, let's move on to
I'm using my phone as we speak now.
Okay. But can the learners hear? No. Okay, let's go to the next school that would be ready. We'll go back to Dr. Petersen later. Which school is ready now? I see there's a learner ready. Which school is this? Nomzamo. Okay, go Nomzamo. Good. Go for it.
Is it necessary to take this if your immune system is strong? And can a person be immune to a coronavirus?
Okay, that's number one. Go to number two. We've got 3 questions here.
Good morning. My name is Yuemka from Nomzamo High School. What is the variant?
Okay, great. Number 2. Now, question number 3, which is the last one.
Greeting, doctors. My name is Azle Papa. I would like to know how to virus mutate itself, and what conditions are required for virus to for mutation to occur.
Great stuff. Perfect. All right, Jack, it's your opportunity.
Okay, those are very good questions. So, is it necessary to take a vaccine if your immune system is strong? Can a person be immune to a coronavirus? Good questions. So again, you can assume that most of us, unless you have something like, say, for instance, you're old, like old people tend to have a very weakened immune response. Same thing with also young children. They tend to, because they've not really had a chance to develop a strong immune system. They've not seen a lot of pathogens in their lives to really push the immune system to develop. So they tend to have weaker immune systems, and also with old people, and again with people with HIV, because HIV depletes your T cells, so it makes you immunosuppressed. So those people tend to have a weak immune system. But everyone else, you can then say, they have a strong immune system, but they still get sick, right? If they're getting an infection with a bacteria or a virus. So even if your immune system is strong, right, and you should be able to clear the virus, but it's going to take you longer compared to someone who's been exposed or who's been given a vaccine. Because what the vaccine does is that it basically educates your immune system to be able to be equipped with the tools that will allow it to fight the virus better, right? Now, although your immune system is strong, it is not specific to the coronavirus, right? So it's going to take time. So what the vaccine does, your body will do when you're getting an infection with the real deal, the virus, right? Whereas if you expose your body to the vaccine, your body takes the time to learn this new foreign pathogen, the what is in the vaccine, build what is called memory. Because a critical part of vaccines is basically not in the immediate response, but it's in the ability of the vaccine to produce what is called your memory cells. So your memory T cells or memory B cells. Because these are the cells that will remember in future that, hey, we have seen this thing before, and when we saw it previously, this is how we responded. We're able to increase the secretion of this antibody. We're able to increase the secretion of this cytokine. We're able to increase all these different molecules that are required by the body to deal with the infection, right? And this is what a vaccine does. So even if you're immune competent, meaning that you have a strong immune system, you still need that education so that you then quickly respond to a future infection, right? Now, when you don't do that, when you don't give your immune system that, exposure to this vaccine, when you do get infected, it's going to take you longer to clear. You may not necessarily be as sick as someone who's immunocompromised, but it's going to take you a while, to deal with the same infection compared to someone who had a vaccine, right? It's like, if you think about it, like I say you have flu in a season, like let's say this year we are now in winter season, right? Of course, we know that every flu season is driven by a different variant of the flu virus, right? Or the flu viruses, because there's many viruses that cause flu. Now, let's say you get a flu at the beginning of winter. Let's say now you get flu, right? If you think about it, the subsequent flu in the same season don't last as long as the first, flu bout that you get, because you're seeing a new variant of the virus. You don't know how to respond to it. It takes you a while, like you're sick for a week. But if you get sick second time around in the same season, you're sick for 2-3 days and then you're back to life, because your system has seen that variant before, and then it's able to respond quicker to the same virus, right? And this is really what the vaccine offers. Although your immune system is strong, it should be able to deal with the virus. It may just take longer to clear the virus. So that's the response to the first question. Second question is, can you be immune to COVID? In my reading of the literature, I have not come across cases where people, have been shown to just be immune to COVID, meaning that basically you can infect them and then they don't develop any, sickness. What we've seen is that people, of course, who would get infected and they don't, get as sick as other people, right? But that doesn't imply that you are immune. It just means that, your body is able to deal better, with the virus, right? I think when it comes to, immune, I think if we look at HIV, I'm trying to remember now the studies. It's been a while ago. There was a group of women, I think they're somewhere in a tribe in Africa, maybe in Zambia, if I'm not mistaken, who had a variation in their T cells that basically, prevented the entry of the virus, right? So these women basically, what you would say is that they are naturally resistant to HIV. Right? They will get the virus can get into their systems, but it can't penetrate their cells because they have this change in their cells, right? And these people then basically are resistant. Like they don't get infected, they don't get sick of HIV because of those changes in their immune cells, right? In COVID, I don't think we've had such reports yet. But I don't really work a lot on COVID, so I don't read too much of the literature on COVID. So I may have missed a paper that speaks about that. I'm not going to claim to be an authority in that regard. But as far as I know, we don't seem to have cases of people who are just naturally resistant to COVID. So we all get sick, we all get infected, but some people don't show the signs of infection, where some people show signs of being infected. The second question was, what is a variant? And then the third question, because I want to combine those two questions: what is a variant, and how do viruses mutate, and what conditions are required for mutation to occur? Because they are interlinked. In fact, I think this third question comes first before you get to the second question. So viruses, basically what they do is that, specifically with COVID, with coronavirus, it's an RNA virus, right? So you know that, when in terms of storing our genetic information, we have what is called DNA as humans, right? In our cells, we have DNA, and then DNA gets transcribed to RNA, then RNA gets translated into proteins, and the proteins perform work in our cells, right? So with viruses, coronavirus is an RNA virus, meaning that basically they don't have DNA, but they have RNA as the source of their genetic information. Now, this is where basically all the components of the virus are contained. They're contained in this RNA, meaning that, what proteins need to be on the surface of the virus, how the virus functions, is all contained in the genetic information, right? Now, this genetic information, when the virus propagates itself, when it divides and multiplies, also this genetic information needs to be multiplied, needs to be copied, basically. And in the process of copying this genetic information, mistakes can be made, right? And this is what we call mutations. So you can remove something, which is a deletion. You can insert something that's not there before, which is an insertion. Or you can have what is called your, you can insert or delete, components of this genetic information, right? In the process of multiplying, or in the process of copying this genetic information. And this is when then you're introducing mutations. Now, different viruses mutate at different rates. So there are viruses that, mutate very fast, and then there are viruses that mutate very slowly, right? So once we have a mutation in that genetic information, depending on where the mutation is, it could be silent, meaning that although you have the mutation in the RNA, it renders no difference, right? There's no difference that you can see. Some mutations can kill the virus, right? So some viruses, so a virus can mutate, and then the product that comes from that can really be like killed because of the mutation happens in a critical component of the virus. Let's say, for instance, in components are required to assemble the virus. If they have a mutation in those enzymes, then basically, your virus is dead. It does not multiply anymore because you have a mutation in a critical component of the virus. And then you have mutations that will enhance the infectivity of the virus, right? And this is what you get like a variant, like what you got in SA with our own variant, right? So this is a mutation that happened on the spike protein that then renders the virus more infective, more transmissible, right? So then you get the Italian variant, you get the South African variant, you get the Indian variant, you get the Brazilian variant, and many other variants. They all come from these random acts of, reading this genetic code and introducing errors in the genetic code that then lead to, a variant or a mutant first, right? That then is more infective. And then once you get this stable virus, then you can then call that a variant, right? It's just a virus that is basically not the same as the parent, the original, right? That then becomes what is called a variant. And variants, basically the conditions that viruses need to divide. And this is why they infect us. They factors because they want to survive. They want to produce more of themselves, right? And in the process of trying to produce more of themselves, they need to copy their RNA. And in the process of copying the RNA, they introduce mistakes. And because it's an RNA virus, there's no mechanism to come back and make corrections. Because the reason why we don't get issues with our cells when we're moving from DNA to RNA to proteins is because there are enzymes that come back and check, did we introduce errors when we were multiplying our DNA as human beings? And when you have errors, then there are mechanisms that come in and say, well, in this cell, there's an error of a mutation. We need to kill the cell. And then basically those mistakes get, discarded. And this is why we're able to maintain the integrity of, of our bodies as human beings. It's because of the mechanism that come to check for corrections, for mistakes that happened whilst we are dividing our DNA. And when you do have issues and you don't get a correction, this is then when you're getting cancers, right? Because cancers mostly come from errors in the genetic code that then allow cells to divide rapidly, and then getting the formation of tumors and all the stuff, right? So checking for mistakes is critical to maintaining the integrity of our DNA and making sure that we remain healthy as human beings. And the same is true for viruses. But of course, for viruses, that sometimes those errors could help the virus become more virulent, become more infective. Let me see if did I miss another? Yeah, no, I've answered all of them, those 3 questions.
Okay, thanks, Doc. I think we've got, for what I see, there's a plan for Nomzamo to ask their questions. Okay? Or rather, Hector Petersen, sorry, my apologies. Hector Petersen, what is the plan for the questions? Are we good with the questions, Hector Petersen? I saw in the message that you were going to use your phone.
Greetings to all. My name is Masibambisane and I am a representative of Hector Petersen. Our question number 1 says, how long will the vaccine last? Can a vaccinated person be determined as safe? Must we wear masks after vaccination?
Thank you.
Question number 2 says, are vaccines 100% trusted for every age? Are there different reactions for people of different ages? Question number 3, in which form is the vaccine? Is it a pill or injection? Number 4, are vaccines costly? How much per dose? What about poor people?
Great.
Then round 2, question number 1, we know that AstraZeneca is one of the vaccines that are ineffective with the South Africa Delta variant of COVID-19. Have any traditional medicine been tested or approved to cure COVID-19? Question number 4, as we know that Indian variant Delta of COVID-19 has arrived in many parts of the world, are scientists going to invest another vaccine for every variant? Question number 3, can a baby be born with COVID-19? Does the baby go with the vaccination program? I thank you.
Okay, I think Petersen took all of the round 1 and round 2 questions in one go. Let's see how far you can go, perhaps, Doc, in trying to.
Yeah, okay. I'm going to very short answers.
Okay,
so
yes.
I'm going to start with question, the last 2 questions. I'm trying to find my questions. Yeah, I'm going to start with question 4 and 3, right? So with question 3, in which form is a vaccine, pill or injection? All the vaccines that we have right now available are in a form of injection. So you need to go and get an injection. In fact, I think most vaccines that we do take in the country are in a form of an injection. There are some vaccines, of course, to take what is called sublingually, where you basically like drops. This is basically you just take the drops orally. But in the case of COVID-19, all the vaccines are by injection. So that's an easy question. The question of cost. Vaccines are very expensive. I think when I was checking the prices, I think for the J&J vaccine, we pay about 153 rands per dose. For the Pfizer vaccine, which I think we're getting some doses of a Pfizer vaccine. In fact, I think we are already using Pfizer vaccine right now in the country. We're paying about 299 rands per dose. And then for the Moderna vaccine, and Pfizer and Moderna basically are same type of vaccine. So both of them are the RNA based vaccines. The Moderna vaccine is about 586 rands per dose. So they're very, very expensive. And this is why, again, when it comes to the poor, government is paying for the vaccines, right? So that, those who are poor and cannot afford can have access to the vaccines. Otherwise, it will become very, very costly. Because with the Moderna and the Pfizer vaccines, people need to go for 2 doses. You need to go and do a booster dose after, I think, 28 days. So you get 2 injections. One is your primer, and then the second one is a booster injection. So that could become very expensive. And this is why, when you think about vaccinating the entire country, it runs into billions and billions of rands. So it's a very, very expensive exercise. And the state is covering the cost, because again, there's a benefit to the states. Is if people get vaccinated, one is you can stop transmission of the virus, which means that people then can go back to work full time and become productive. And then how you recover the money to spend in a vaccine is by productivity at the workplace. When people become productive at work, they are safe, then you're getting your taxes, then your GDP grows, and then the state makes money that way. In terms of question number 1, how long does a vaccine last, and can it be determined to be safe? Well, I think for COVID vaccines, what they say typically in terms of and again with vaccines for viruses, because viruses, they tend to mutate. Usually you're getting efficacy for 6 months to a year. Excuse me. Then every year you almost need to basically get a new vaccine. And hence why for flu, we always have to for flu, you need to get your flu vaccine every year. And I suspect that for COVID, it's going to be the same thing, where we're going to get this yearly vaccination for coronavirus, right? Which is the same thing to do for influenza, that every year you need to go and get a flu vaccine. But in terms of other vaccines that we take in general, it really varies from vaccine to vaccine. We know, for instance, with the TB vaccine, that is very effective when you're younger, and as you get older, it tends to lose its efficacy. And this is why we even now, although we have a vaccine for TB, which is called BCG, we still need more vaccines that are going to be effective even for other people, beyond the teenage years, right? So yeah, it's not that a vaccine, once you take a vaccine, it's going to be safe for a lifetime. Some of them tend to wane over time. With COVID vaccine, I think what has been shown is that the vaccine are effective for a year. But again, I think studies are ongoing to see how long. So if you look at people, for instance, in countries like US who got a vaccine last year, then we can see whether they will be protected after 2 years and 3 years and 4 years and 5 years. Because really, we don't have the data, because we've only been giving these vaccines for a year and a half at most. So until we look in 5 years' time and say what happens to these people who got a vaccine 5 years ago, are they still being protected? We're not gonna know, right? So at this stage, really, it's just speculation. The other question was about once you have taken a vaccine, are you not gonna get, are you safe? And the answer is, again, is no. This is why even people who've had a vaccine continue to wear masks. is because, and as I said earlier when I was answering one of the questions from Nomzamo from Masibambisane was, the vaccines don't prevent infection, right? They prevent severe disease. So although you've been vaccinated, you're still going to get sick or not, you're still going to get infected. If you do get sick, you're not going to be severely sick, right? So you may not need to go to hospital, but you're going to have a flu, a fever and all those things, right? And hence why we still basically advocating for people, even after they've been vaccinated, to continue to wear the mask to prevent infection, to prevent spreading, the virus. Another key component that you don't know as scientists is that once you've been vaccinated and you get infected, although you don't get seriously sick, but can you spread the virus? And this is critical for transmission, because ultimately what we're trying to do with the vaccine is to cut the transmission, right? So when a person has been vaccinated and you don't wear a mask, you get infected. Although you may not get sick, like you don't get too sick, but we don't know whether you can be able to transmit the virus to the next person. And if you can transmit to the next person who's not vaccinated, then it defeats the whole point. Hence why, again, even though you may be vaccinated, you may still need to wear a mask. And I think masks are going to be with us for a very long time when it comes to COVID. The good thing with masks, which maybe we don't really discuss a lot, is that if you look at the incidence of things like flu, it has gone down as more and more people are wearing masks, right? And you'll see, like in school, teachers can observe how many learners had flu, like in the previous years, compared to last year and compared to this year. The numbers are going to go down because, again, now everyone, like if you get flu, you sneeze, you cough, now everyone is covered. That transmission of the flu viruses has also been cut through trying to prevent the spread of COVID. So it's an unintended positive benefit of wearing a mask. The other question was, are vaccine 100% trusted for every age? To answer that question, again, you need to go back to the clinical trial stage, right? Is when you're developing a vaccine, you need to make sure that the vaccine is safe for every age group, right? So you don't only just recruit, let's say, people who are between the ages of 18 and 24, and then from there, extrapolate that the vaccine is going to be safe for someone who's 60, right? So in the sample of people who are, undergoing the clinical trial phase, you have all ages of people, right? You have young people, you have middle-aged, you have old, you have very old people. Because we want to be sure that for every age group, the vaccine is safe, right? And it's part of the testing process. Because if you test a vaccine, but you only test it in one small, age group of people, then basically it's not going to be approved for use for older people, right? And again, I think with there was issues at the beginning. I think it may have been the AstraZeneca vaccine where it was issued around pregnant women. But then when they did bigger clinical trials, they found that actually there was no problem. And this is why even for pregnant women, they can take, the COVID vaccines because they've been shown to be safe even for pregnant women. And this is all the checks that need to be done, because you can't assume that because, the vaccine has been shown to be safe in men, it's going to be safe in women, right? Again, you can't assume because it's vaccine has been shown to be safe in 20-year-olds, it's going to be safe for 50-year-olds. So all of that needs to be checked, analyzed, and then approved accordingly. So in terms of COVID vaccines that we have now, they've been tested in all age groups, and they've been shown to be safe, hence why they're given for everyone, including the people who are older, like what we're doing in SA right now, where we're giving the vaccines to the 60 and above people.
Okay.
So we can go for round 2, right?
Yeah, in fact, there's one other school, Oval North.
Sure.
They in the house, I'm not sure. Oval North, are you there for your questions?
Hi, Mr. Yaga. Good day, colleagues. Oval North is not in the stream today. They had some issues.
So
I don't know if we can ask the one question on behalf of them.
Okay.
Dr. Ndlovu. I know we're running fast out of time.
Okay.
Perhaps I think you've touched on it in terms of the development of the immune response. So if you have had COVID or you know, some kind of disease, and then you get the vaccine, could those two T cells, could there be a difference between them? And how do they coexist if there is a difference? You know, so if you get a immune response from your body and then you produce another immune response from a vaccine, is there a relationship between those two?
Yeah, sure. Okay, so basically, you know, how this works is, so in immunology, basically what you learn is that T cells are blind, that they're completely blind, right? So for T cells to be able to respond to any pathogen, any infection, they need to be educated to respond to it, right? And this is why then you get what you call your cells of the innate immune system, which have an ability to engulf the bacteria or the virus. They're able to kill it, break it down into small components, and then take the components of the virus or bacteria and then present it to T cells and educate them. So how T cells see this new information that is being introduced to them is via what is called a T cell receptor, right? So this is a receptor that is sitting on the surface of a T cell that then goes and recognizes this small molecule or piece of the virus or a bacteria, right? And then they become specific to responding to that, you know, pathogen, that piece of the virus or bacteria that they saw, right? Now, in our lifetime, we see a lot of pathogens, right? So we have T cells are specific for many different things. You know, T cells are specific for TB, for parasites, for whatever, right? And they all just get stored. So that memory gets stored elsewhere, and there is no conflict between all these different types of T cells, right? It's just a matter of how they were educated. They just go there and sit in the place where they need to sit until they are required to come and respond to a particular infection. And this is really how it works. So there's no conflict, because what is changing basically is that bit of the T cell that recognizes the antigen, right? Which is the receptor, right? And in terms of the T cells, we make a lot of T cells, right? We make tons of T cells. In fact, if we to go and look at organ like the thymus, which is where T cells get really made, every day we produce a lot of T cells, and all these T cells have different T cell receptors, right? So they can respond to as many pathogens as possible, right? So they're not specific to one pathogen. They only become specific to one pathogen after they've seen the pathogen, right? And this is how it works.
Okay.
Thank you so much, Tom.
All right, let's agree. We are at 12 o'clock, and what we will do, we will rush through round 2, if it's okay with everybody. So we're back to Masibambisane. Are you there for your round 2 questions? Masibambisane, are you there for your round 2 questions? Okay. If not, Nomzamo, step in with your round 2 questions. Okay. Who's ready to come in?
Greetings to Dr. Hlumani. I would like to thank you for the feedback you gave us first.
Okay.
So, I come from Masibambisane High School in Delft. The information that you gave us first, it was overwhelming, and we thank you for that. It was a great honor on learning how the vaccine worked during this COVID time. It was very helpful, and thank you for that. Now, we'd like to ask our second question.
Good morning, Mr. Ndlovu. And the question I want to ask is that you spoke about a parasitic worm that infects humans and causes an immune response. What is the name of the worm, and how is the infection caused by the worm? What is the symptoms in the human body caused by the parasitic worm? That is my question.
Thank you. Question number 2, Masibambisane.
Our next question is, what other careers are available in the field of biochemistry?
Okay.
Sure. The name of the worms, quickly. I mean, I work on worms called Schistosoma mansoni. So they cause a disease called bilharzia. And basically, you know, the symptoms vary. But in the main, what you see is you get fever typically, and also you get, you know, people tend to lose weight. You know, because, you know, I mean, the worms typically affect the liver, and the liver is the energy machinery in our bodies. So it's fever, it's weight loss, and many other symptoms. In terms of careers for biochemistry, it really is unlimited, you know, in terms of where you can go. You know, I've had people like myself. I mean, I'm an academic now, so I've gone as far as a PhD, and now I teach and do research at university. You can also go and work in the biotech industry, where you can develop vaccines, can develop therapies for disease and many other things. You can also take your biochemistry degree. You can go into IP law, where you can then, you know, take your knowledge of science and apply it in law. Of course, you need to do a couple of modules in law to qualify as a lawyer, but then you then use your knowledge of science in terms of how, you know, innovation and how it works, and then how then to try and use law to protect those who develop ideas for, you know, society. You can work in politics with a science degree. You can go and work as a consultant in these consulting companies. Really, I mean, because ultimately what we try and teach, and this is how I explain it to my students, because they always come back and say, well, I'm going to do my master's or PhD, where do I go? Is what we try to teach you in science is how to solve problems. And that's really how you should see it. Don't see yourself as a biochemist or as a geneticist or whatever. See yourself as someone who has the skills to solve problems. And then you go outside into you know, the work environment, looking for problems and trying to find solutions. And you can go anywhere. I know guys who are now consultants. I know guys who've qualified as biochemists now. They are in finance. You know, you can really take it anywhere. It's just a matter of training your mind to basically solve problems. So that's really a quick answer to those two questions. I know that we are running out of time, so I'm not going to explain a lot more about parasites and how they work. Sure.
Thanks, Doc. Nomzamo, round 2 questions. Nomzamo. Do we have Nomzamo ready for round 2 questions? Okay, I see there's a learner there ready to speak. Is that from Nomzamo?
I would like to ask the question: if how long does it take to manufacture the vaccine?
Okay. Number 2.
Good day, Doctor. I'd like to know how do the pathogens in the vaccine weakened it?
All right. Number 3.
Good day, doctors. My name is Viewer from high school. I would like to know, is there a difference between the vaccines being used in South Africa or in other countries?
Thank you so much.
Yeah. Okay. I'm going to start with the last question because I think it's the easiest one to respond to. The vaccines are the same. They're made by the same companies. So we're getting the same vaccine that was used in the US in terms of the J&J or the Pfizer or the Moderna vaccines. They are not different. It's like, I mean, I think they're trying to manufacture the J&J vaccine in PE because Aspen has a license from Johnson & Johnson to do some manufacturing. But I don't think the vaccine is being used locally right now. So I think the batch that we have are coming from overseas. So we're using the same batch, I mean, or same vaccine as what is being used elsewhere in the world. So there are no differences. I mean, they are made by the same companies using the same technologies. It's exactly the same. And they go through the same approval process as well. You know, in fact, I think our regulator for vaccines, SAPRA, tends to rely a lot on the US FDA, which is the US Federal Drug Administration Agency, which of course has a responsibility to approve new treatments and vaccines. So they get a lot of their data from them to say, we gave this vaccine approval because of A, B, C, D, and that's how then they're able to then do the same thing here in South Africa. So there are no differences. It's the same vaccine that is used in the US, is used in SA, is used in India, is used in China. The other question was around, let me just confirm, how is the pathogen weakened? So there's many ways to weaken the pathogen. One of which is to use heat, right? Because once you learn, again, if you go back to in biology, you learn about proteins, and proteins are, highly sensitive to heat. So if you expose your virus to high heat, because I mean, viruses basically they function in our body. And our body temperature is about 37 degrees, right? So if you were to expose your virus to like a temperature, let's say of 56 degrees or higher for like an hour, you basically are killing that virus, right? So you have what is called heat-killed, viruses that you can use in your vaccine. And in the case of COVID, what they use what is called chemical treatment. So basically use a chemical to kill the virus, and we basically render the virus unable to replicate. And if the virus cannot replicate, it cannot cause sickness. And this is how then it's another way of trying to weaken the virus. Another way is to then also mutate the virus, is to remove the component of the virus that are driving its virulence. And then in that way, then what you get is a mutant that is weaker than the parent. So there's like genetic, chemical, or using heat to weaken the virus. The other question was, how long does it take to manufacture the vaccine? Now, I'm not sure whether you mean to develop the vaccine until you have a full vaccine, or you have the vaccine, now just need to continue to produce the vaccine. Because if you're asking about like, how does Pfizer or Johnson & Johnson produce the vaccine, how long does it take? I have no idea. I'm not going to lie. I think the companies have that information. I don't have the information, how long it takes them to produce one dose of the vaccine. But in terms of like developing the vaccine, what we know is that it has taken what? I think what was able to assist in developing vaccines for COVID was because we've had outbreaks of similar viruses before. So companies had already been in the process of developing vaccines for other coronaviruses, right? I think what we had in the Middle East, what we had MERS. MERS is Middle East, respiratory syndrome coronavirus. It's caused by also coronavirus, but then it went from camels into humans. And then if you remember in China, I think around 2003, there was a time when Chinese were wearing masks and they had an outbreak of SARS, right? And again, it's another coronavirus that was causing that outbreak. So we've dealt with them before. We're in the process of developing vaccines for those coronaviruses. So we use the same backbone to develop vaccines for coronavirus, the current pandemic that we have now. And hence why we're able to move quicker, because we already had the technology trying to apply it into different coronaviruses. The same also technology is being used currently now to produce a vaccine for Ebola. Again, I think you guys will be aware of Ebola because it's been causing problems in West Africa specifically. I think we've had an outbreak this year in, I think, Congo. There was an outbreak like a month or two ago. where there's an outbreak of Ebola. And the Ebola vaccine is built the same way as the J&J vaccine. J&J and AstraZeneca vaccines have a similar, makeup as the Ebola vaccine. The difference are just the antigens that you're targeting to induce immune response.
Okay. All right. As we wrap it up, Dr. Ndlovu, wonderful interaction with the schools. Very, very interesting questions from everybody. And I would like to give the opportunity to the educators to say a word or two before we close it down, because I think we've exhausted more than an hour and wouldn't like to get stepped too much into the second hour. So can I get the teachers in the order of Masibambisane, Hector Petersen, and Nomzamo to say a word or two to everybody? Doesn't have to be long, it can be just a few seconds. So Masibambisane, Mem Kubando, a word from you.
Yes, Masibambisane, teacher is ready. Yes. Good afternoon, Doc and the team. I would like to thank Ithemba Labs for giving us this opportunity to meet with you, Doc, and also we're talking about the very important topic, which is the vaccine, which we are under the COVID-19 pandemic. So our learners were very excited for this day to come, and they are very pleasant to be with you and talk to you and pose the questions. And they've really learned a lot today. Even myself, I've learned a lot about a lot of things. And also, you give them a chance now to make an effort in doing physical science and maths as they are doing. So they are also interested to pursue with doing the biochemistry. So thank you very much for your input. And Ithemba Labs, you always bring in the good topics for science learners. Thank you very much for that. Thank you so much to the team and everyone. Bye.
Thank you so much. Thank you so much. Thank you so much, ma'am. Our appreciation to you as well. Thank you so much. All right. I said, what's the next one? Ma'am from Nomzamo. Is there somebody from Nomzamo to say thank you? Nomzamo.
Good day, colleagues, to the presenter and the doctors available, the colleagues. I would like to thank you for the presentation. Really, as Nomzamo High School, we didn't even, I'm sure the learners also didn't even know about the vaccine, but at least now they have enough and they can motivate people to go and do vaccination on this product. I will allow one of our learners to also say something. Thank you.
Okay, thank you, ma'am.
Good day, Doctor. I find it very interesting that I learned a lot about the vaccine and the virus, all of the stuff that you have mentioned above, and I think the opportunity that you gave us to ask the questions so that we can learn more. Thank you.
Thank you, guys. Lastly, Hector Petersen, ma'am. Hector Petersen. All right. Is that an indication? Okay.
Okay. Good day. Yes,
So,
it's an indication that Hector Petersen wants to speak now.
Okay.
I would like to thank Dr. Hlumani Ndlovu for what he experienced from the beginning of schooling up to at the moment that he's a researcher, because that is a stimulation to our kids, that is an inspiration to our kids, because they usually take one channel that is already in influx. So now they've got a broad mind that are going to take some, become immunologists, that is an interest to them. They are going to be virologists, that is an interest to them. Obviously, we need more people that are going to be in a science lab. So they want to be the people that are going to be on that line. And I thank you, Mr. Tobias, for helping us with the connection. We were struggling, but you made us to calm down. I thank you so much. And the team that is working with us at Ithemba Labs, that communicated us so well, that makes us to calm down and be connected as well. And I thank the other teachers that we engaged. Thank you, Nomzamo. Thank you, Gordon High School. Thank you, Masibambisane, our neighbor school. And we wish to have a team that is going to be big, producing scientists in South Africa. We need more. Thank you. Over to my learners that are going to make a sound of a noise.
Thank you.
Thank you, guys. Thank you so much. All right.
Do you want to say something about the inspiration of these?
I would like to say thank you, Dr. Hlumani Ndlovu. You taught us a lot. As we know that in the beginning of grade 11, we learned about viruses and bacteria. So we would like to thank you so much for the information that you gave us, the broad information. I thank you again.
Thank you very much, guys.
To let Masibambisane off the hook.
Thank you once again.
No Lena from Masibambisane. Now that others have had Lena say something. Masibambisane, just one person, 2 words, 3 words. Okay. All right, it's fine. Perhaps there are some issues, but overall, people, thank you so much, everybody. It's been a wonderful, wonderful session. One of the best ever. Ma'am? Okay, sorry. One of the best ever, and I definitely will continue on this path. We will keep on bringing you topics that speaks to your way of life, topics that resonate with you, because we feel that that is the only way we can get our learners to basically appreciate math and science. It speaks to us. It speaks to things that we do. It speaks to the way we live. It's just that perhaps we just miss, introducing it to you the way we're supposed to. So this is one way of us trying to get you more involved, more enthusiastic about it, more getting into love and getting passion of the subject. So thank you so much to everybody. Thank you, Doctor, for your time. And you really are a very special speaker. I must say, we've had a lot of them, but you are one of a kind. And you really can relate to our people. Thank you so much for your time. We appreciate it. Mr. Tobias, closing remarks as your parting shot.
From my side, colleagues, I think everybody, it was such a pleasure getting ready for today. We are really thankful to all of the schools that have joined us. We know that it is a struggle to put together some of these things within this uncertain time. But the commitment and the effort shown by the educators at our schools, it's just one of the things that I want to just take my hat off and then just thank them. And they sit in the middle of my heart. I'm really happy to have worked with them today. Thank you so much, Dr. Ndlovu, as well. Mr. Yaga has pointed out, everything. You have provided us with so much, with a resource of information, a really large resource, and we appreciate that. From my side, yeah.
That's maybe as a parting shot on my side. I mean, it really is a pleasure for me to get this opportunity to interact with young Black, learners. I mean, we don't say that enough, because I think these learners are the generation that, we are waiting for, in terms of taking this country forward in many respects, particularly in the frontier of science and innovation. So I really want to encourage them to really, grab this opportunity with both hands and really push forward. I think, in the past, in this country, there's been many barriers that try to limit the excellence of Black people in terms of specifically in the fields of maths and physics and the sciences in general, right? Now we know for a fact that we are very smart as a people, and nothing should stop us. No one should question your ability to comprehend and do mathematics or physical science or chemistry. You are the person that you think you are, and you guys are amazing. And just go out there, believe in that. Know that you're going to be a doctor, a scientist, an engineer, whatever that you want to be in life, and nothing is going to stop you. If you maintain a self-belief in yourself, nothing is going to stop you. So I wish you guys all the best. It's a pleasure. I hope to see you guys in the next few years at UCT, and I'm teaching you guys medicine at first year level. or third year, or maybe see you in my lab as my students and doing TB, research in future. Thank you for the opportunity to the Itemba Lab team. It's really, for me, I like talking to young people, so I'm always available. Thanks for the opportunity.
Thank you, Dr. Ndlovu. Thank you so much, guys. With those wonderful words, we are closing it. Have a good day, everybody. Thank you so much for connecting. See you next time.
Yes.
Sharp, sharp, ciao.
See you, bye-bye.
Right.